episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome everybody again to another episode of our show, bringing you another fascinating guest, helping to create a better tomorrow on many different fronts. Today we have the honor of being joined by Dr. Morgan Levine, who is Assistant Professor of Pathology and Epidemiology at Yale School of Medicine. Dr. Levine is also a member of both the Yale Combined Program in Computational Biology and Bioinformatics, as well as the Yale Center for Research on Aging, and her work relies on a fascinating interdisciplinary approach, integrating theories and methods from statistical genetics, computational biology, mathematical demography, to ultimately develop biomarkers of aging for humans and animal models using this high-dimensional omics data. As a primary investigator or co-investigator on multiple NIH, Foundation, university-funded projects, she has extensive experience at using systems-level machine learning approaches to ultimately look at epigenetic, transcriptomic, and proteomic changes with aging, and ultimately incorporate this information to develop measures of risk stratification for major chronic diseases, including cancer and Alzheimer's. Her work involves the development of systems-level outcome measures of aging aimed at facilitating evaluation for gero-protective interventions, and a number of the existing biologic aging measures that she has developed are being applied in both basic and observational research. Dr. Levine has her PhD from University of Southern California in gerontology, her postdoc work from UCLA. A lot to talk about. Dr. Morgan Levine, thank you so much for taking the time to come join us today.
Thank you for having me.
It's really great having you. I'd love to start things off, just as we typically do, by handing you the floor for a couple of minutes to just talk a little bit more about yourself, a little bit about your background, from everything from where you grew up, how you got interested in science, and ultimately a little bit of your path into the field of aging. I think that'd be a great way to start things off here.
Yeah, so I grew up in Los Angeles. So until actually I moved to Yale, I lived in Los Angeles pretty much my entire life. I try to think back when I became interested in aging at first, because I was interested in it for a very long time. And I think it was always because when I was born, my father was actually quite a bit older than average. I think he was 54 years old. So aging was something I was always worried about growing up, something I always felt like I wish I could control or slow down somehow. And then actually, my mother is also an academic, so she actually works in aging, but on the policy side, so things like long-term care. So I think it was a little bit of a natural thing, and I just did my undergrad at USC as well, and through that became affiliated with the gerontology program there, which is really cutting edge for both social science and biology of aging research. And they offer a very multidisciplinary kind of program in focus specifically on aging. So I ended up doing that PhD and just kind of continuing on trying to figure out how we can take a little bit more control of how each of us ages.
Excellent, excellent. And a core of your laboratory focus is in the area of epigenetics. And one of the fascinating things when I listen to some of your past presentations and sort of read through your research is that you really come at epigenetics from two very interesting angles. You come at it, one from sort of the molecular biology perspective of what we've spoken about a little bit about in the show in the past, that looking at the DNA methylation, histone modifications, things at that level. But then you also talk about another topic in the epigenetic space, which you don't hear as much about, but this sort of more metaphorical epigenetic landscape, originally sort of coined Conrad Waddington, but now it's coming a little bit more back into vogue in terms of the systems biology folks that say, okay, this is not just a metaphor, but really we have to look at sort of this, sort of these networks across entire systems and how all this epigenetic stuff is playing out. And I'd love to, I guess we start with sort of the molecular biology biology perspective. You're very active in the area of epigenetics from the perspective of epigenetic clocks. You give very long and detailed presentations on this topic. Can you take us on just a little walk through sort of where you are in 2021 with regard to the development of these different clocks? What are some of the things that sort of we understand in September 2021, and sort of where you're going with some of the epigenetic clock research today?
Yeah, so I mean, I became very interested in the epigenetic clock field just because to me it seemed like this magical phenomenon. So something where you can measure the exact same sites across almost any tissue and it tracks with age. And to me, I was just, it's a great tool, but I also became very interested in why does this happen? What is this coming from? What does it mean? So I think really that's where the field is going to now. We've shown we can develop really good epigenetic clocks. We can develop very good age predictors where we can estimate age within a few years in a variety of different tissue and cell types. We're also developing better predictors of aging-related outcomes. So whether it's remaining life expectancy or health outcomes. But I think what we really lack now is kind of a mechanistic understanding of what these patterns that we're putting into these algorithms actually represent, which ones are meaningful to track and measure, and how they relate to each other. And so I really think moving forward, that's where the field is going to go, is trying to understand whether it's using more in vitro data, more animal models, where you can do kind of experimental manipulation to understand what the different kind of changes in methylation actually mean and what other phenomenon they relate to and reflect.
And then transitioning that from sort of the molecular bio level now into what's termed in sort of your laboratory sites definition, sort of these cellular aging trajectories, and now we're coming to sort of the epigenetic landscape component of this. I heard you you talk recently. You were talking about sort of with the epigenetic clocks. A lot of us could have similar epigenetic clock readouts, but we get there via different ways. And it got me thinking a little bit about sort of cancer in a way. We could have many different mutations that lead to the same cancer cell, or certain cancer cells that have different types of mutations and so forth. As you start thinking about the sort of where epigenetics and sort of the epigenetic readout per the clocks leads us in terms of what happens next, whether that's a differentiation of cells or cancer formation, or as the systems bio people think, these attractor states of disease or health or what have you. What talk about a little bit about the road you're going down on that path, because that's equally fascinating to sort of the development of the clocks, in my opinion.
Yeah, I think we really need to understand kind of the states that these things basically represent. So people are always interested in, which genes are you finding, differential methylation in? But it's not like a single gene that's getting up or down regulated. It's this pattern. And when we talk about kind of the landscape, the question is, what does, the pattern of a cell or a tissue or, whatever we're measuring this in, how does that define the state of that entity? So, and how did that pattern change, or why did that pattern change to kind of get there? And can you move along this landscape? So can you change your state by changing your methylation, whether that's through some intervention, or we also know just with aging, your state is going to change. And what does that mean for kind of the trajectory you're taking along this landscape? So in my lab, we're not that interested in specifically the individual CPG sites, even though, they all go into the clocks and you have a few hundred of them or 1,000, depending on which clock you're looking at. But it's more at kind of understanding this really complex code that's in the epigenome. So I like to think of it as like the operating system of the cell. So so kind of your epigenetic pattern of a cell is dictating some structural characteristics of the cell, how much it's replicating, its stress response. So I think the next step is linking these multidimensional complex patterns to cellular states. And I think that's really going to be able to be facilitated through kind of this single cell work that a lot of people are moving towards. It's been a lot trickier with methylation compared to things like gene expression. So single cell RNA-seq is pretty standard and everyone's using it now. And methylation, there are definitely a few groups, but the data is not quite as robust and the techniques aren't quite as kind of agreed upon as RNA-seq. But I think as we get there, it'll hopefully highlight a lot of things that will help us kind of link the pattern to the phenotype.
And as far as, because I mentioned in the intro, obviously epigenetics, a very hot area. You're extremely focused on it, but you also bring in a lot of the work with transcriptomics, proteomics, maybe other omicses. Talk a little bit of how, because obviously there's many different types of clocks that we can follow. Epigenetics, of course, being one of them, but there's folks that focus on other areas. How are you integrating some of these other omics clocks into what you're doing with the epigenetic clocks?
Yeah, so I think the next frontier is really the multi-omic kind of profiles of either individuals or cells or tissues. So how do we integrate these readouts of a bunch of different processes? So yes, right now there are proteomic clocks, there are glycan clocks, there's methylation clocks. Even though I work mostly with methylation clocks, I acknowledge that they are not capturing everything we want to capture about the aging process. They're just showing you a sliver. So how do you combine all of these things, which are hopefully, at least to some degree, independent? So people have always been kind of fixated on, are the proteomic clocks, they want them to measure the same thing as the epigenetic clocks or, the metabolomic clocks. And, they say, oh, there's a problem, they're not giving you the same readout. I think that's good because that's more useful information that we can kind of put together to give us kind of a more multidimensional picture of aging. If we just got redundant information, that it would not be as useful. We'd kind of be reaching our kind of threshold for how much we can kind of model using this type of data. So I think, yeah, the next step is to combine all these different types of, whether it's omics or other types of data. I think that's been a little bit slow because typically you don't have them in the same sample. So in a lot of samples, I'll have methylation, but I don't have proteomics measured in that same sample, or I don't have sometimes RNA-seq. So as people start developing these data sets with kind of these layered omics, then I think it will really be able to kind of capture a more full and complete picture of what's going on in aging.
You're also very interested in the area of epigenetic programming, which has become extremely hot lately. Obviously, it's being sort of divided up nowadays. It's not sort of pure dedifferentiation reprogramming anymore. We have the partial reprogramming folks. We have people working in transdifferentiation to hop between these different cell fates. You were one of the authors on the 2020 paper, Reprogramming to Recover Youthful Epigenetic Information, Restore Vision, with a bunch of other really fascinating people from the aging space. Talk just a little bit about sort of where we are in terms of connecting these various epigenetic reprogramming tools, whichever they may be, there's a bunch out there, with some of what you're learning now in terms of these clocks.
Yeah, so to me, this space is very exciting. Maybe I'm not as excited about it as a translational space yet. I mean, I think there might be potential in the future that might pan out, but I think just from a kind of fundamental basic science question is that we can actually intervene and, like when we were talking about the landscape, move a cell from one spot on the landscape to a completely different spot and change the state of that cell. And we don't know to what degree we can do that, and if it's truly, like it was when it was an embryonic stem cell, or if it's, still slightly different. But I think this is also a great opportunity to start to understand how the epigenetic pattern is mapping onto state. Because if we can actually manually change the state of the cell, then we can look at the epigenome and say, what is the pattern that underlies a cell that's, moved back up this kind of landscape to having a pluripotent kind of phenotype versus, the other questions we're really interested in are, do cells move back up the same trajectory they take when they age or when they're differentiating, or do they move on the landscape to a totally different area or take a different path? And same thing, if you were to age that cell again, would it just continue kind of up and down, back and forth along the same kind of path? Or is it markedly different by kind of this reprogramming? And same thing with kind of this partial or cyclical reprogramming, is how far up do you get? And we're also very interested in, are there kind of barriers or bottlenecks that we actually think we should be able to exploit? So we don't see them, we actually would like to find things that prevent the de-differentiation, but still allow kind of this, what some people call this rejuvenation or regeneration of the cell. But yeah, I think it's a very exciting thing. And I think, prior to a few years ago, no one even knew that this was actually possible.
Mm-hmm. Very recently, you published a paper entitled "Aging Biomarkers in the Brain." Your lab is focused on Alzheimer's disease, but you've also published in the past on other sort of cognitive impairments, schizophrenia, things of this nature. Obviously, the brain is a really unique microenvironment in its own right, and you highlighted in this paper sort of like the numerous interconnected processes that are going on. Once again, talking about epigenetics, proteomics, you talk about neuroimaging, age predictors in this work. Talk a little bit about what you're dealing with in terms of the aging brain, obviously disease, and some of these newer technologies that you're developing and how they can help us further understand this really crucial system that we don't have a lot of answers for, especially when we're talking about things like Alzheimer's disease.
Yeah, so this is actually one space where we are trying to do more of these kind of multi-omics approaches that I talked about. So we currently have a project where we have samples from brain tissue from postmortem individuals, where we're measuring DNA methylation, proteomics, we're looking at somatic mutations and doing lipidomics to really understand how these different things relate to each other. And for that project, we were really interested in kind of this idea of resilience. So we're oversampling individuals who have the ApoE4 genotype, with the idea that about 60% of them are going to go on to develop Alzheimer's in their lifetime. But what is it about the other 40%? Are they just dying of something else when they're still preclinical, or are they somehow resilient? And can we kind of understand and identify kind of the molecular patterns associated with people who are perhaps resilient versus more vulnerable, and use that to kind of understand a little bit more, maybe mechanism of what's actually driving Alzheimer's, because that's, I think, a big missing kind of piece in the Alzheimer's field is, you have people who say it's all about amyloid versus, tau, and I think we don't really understand what is kind of driving the cognitive impairment that we see. We've also become really interested in kind of senescence in the brain. So we have a project looking at kind of interactions between A beta and senescent cells, where we're actually looking at, does the presence of different A beta oligomers actually drive senescence in certain cell types, versus what is it about kind of senescent cells that might change kind of these neuropathologies that you see in Alzheimer's disease. So a lot of this work we're kind of taking on in vitro, so doing kind of cultured brain cells, hopefully organoids in the future, but so far we haven't kind of moved that far into it. But yeah, just trying to understand the complexity of a disease that is fairly poorly understood, despite being studied for a few decades now.
How as I mentioned in the opening when I was talking about your bio, you've had multiple projects, NIH funded, various university funded projects. As a primary investigator, co-investigator, leading these labs. Top line on sort of the current state of aging funding from the perspective of a researcher hanging out at Yale nowadays. What do things look like in 2021?
Well, definitely the Alzheimer's disease funding is very good, as most people who are applying to NIA know, and that's the majority of the budget right now. I do wish there was better funding for kind of basic aging research, because I think that is very informative to both Alzheimer's disease, but also other chronic diseases like cancer, heart disease, diabetes, you name it. So I do wish we did have more kind of basic science funding at NIA. I will say, though, the really exciting thing is the amount of funding coming in from non-government entities. So whether it's private individuals or more of these kind of foundation type grants, I think that is actually a really exciting shift in aging, where, you know, the public and also just biotech in general is getting very interested in the field. And I think that will have a huge impact on maybe enabling more kind of high risk, high reward studies that are not traditionally funded at places like NIA. And I do think it's a good time for people getting into the field of aging in terms of just the funding and the momentum.
Excellent. And your top line opinion, obviously, you know, you're in the core of everything that's happening in terms of epigenetic clock development and these really advanced tools. But, you know, people may listen to the show, they're going to Google this stuff and they'll see stuff for sale nowadays. And top line input on the folks that are trying to rush to commercialize this stuff nowadays, not ready for prime time.
Yeah, so I think it's a question of, you know, what are they going to be used for kind of in the consumer-based market. So I don't think epigenetic clocks are going to like open up a wealth of information for someone. I think they are a good general indicator of how you're doing in terms of your relative health to other individuals. But the same as like getting on the scale every morning, right? So you can weigh yourself and that number doesn't tell you, you know, in 10 years, you're going to get diabetes, but it does give you kind of a baseline for evaluating how you're doing on a day-to-day basis. So I do think the consumer-based market for epigenetic clocks or these other biomarkers are useful for people who just maybe want another piece of information about their health. But I don't think that people should base their entire life around that number that they get. The other thing is that, you know, there's a number of different companies that are providing these, and they're going to probably all give you a different answer. So, you know, again, how much faith do you put in that number? For me, I think the more important thing is, you know, if people can afford it, which unfortunately it's fairly expensive right now, is to kind of track over time, because I actually think that that rate of change is more important than just saying my biological age is X today. It's more, can I somehow see a slower rate of accumulation of biological age relative to chronological age? But again, I think we're just starting to kind of get to that point where people are tracking those things, and the clocks are continually getting better as well. So, you know, whatever's available today is not going to be what we have forever.
Sure. Morgan, we talked about epigenetic clocks, cellular trajectories, epigenetic reprogramming, and obviously the basket that is CNS aging, Alzheimer's, dementia conditions. Obviously, funding is not infinite, but if there was a tremendous amount of funding coming down the pike, what other areas excite you? What else are you interested in? Other technologies, areas of aging science that you know, Morgan Levine Lab #2 would be focusing on?
I mean, for a while we were very interested in kind of the senescence field. I have a few people actually in my lab working in senescence, but that's a very hot topic that, you know, a lot of people are interested in. I'm also very fascinated by parabiosis. Again, not as a therapeutic per se, but as just understanding how, you know, circulating factors or kind of the microenvironment, again, can change the state of cells. So those are kind of the areas we're really interested in. I know, you know, there's a lot of people working on interventions, so rapamycin or rapalogs. They're a little less in my wheelhouse, but I think very exciting, kind of, if they end up panning out through some of these clinical trials that are, seem to be happening first in companion animals and then maybe eventually in humans. And then, of course, as always, the kind of dietary. I know they're old school and people are a little bit bored by caloric restriction and these kinds of things, but I still don't think we have a very good understanding of kind of the molecular responses to these things, and more than that, kind of the different personal from more of like a personal intervention thing. So why does one person physiologically respond one way versus another, or even looking across strains of mice, they don't all benefit from caloric restriction, and some actually have shortened lifespan. So what is it that happens that kind of creates this heterogeneous response, and can you actually use that type of data and information to, in the future, perhaps personalize kind of intervention protocols for individuals based on some kind of molecular profile that they have?
I gotta tell you, if caloric restriction is ultimately what's going to do it for, I think I'm going to be aging because I have a special relationship with food. But anyway,
there's a very few people who can truly maintain a calorically restricted diet for the long time.
Whenever I listen to people talk about that, I'm like, please don't let that be the answer here.
If that's the only answer, then we'll just
move on, do something else.
Just do what we're doing.
Yeah. While I have you, obviously, you've as you've been developing these tools and technologies, you've no doubt worked with a bunch of fascinating people across government, academia, from the industry. Take a little time. I'll hand you back the floor. Any particular mentors, influencers, your team, anyone you want to shout out to, please have the floor on that one.
I've been very lucky throughout my career to have amazing kind of mentors and people I've learned from. So besides, I worked with, just even in my PhD, a lot of really great people in the field of aging. So actually, Eileen Crimmins, who's more on the demography side, but also Caleb Finch, so Tuck Finch, Walter Longo, Hasi Cohen, a lot of amazing people at USC. And then obviously when I did my postdoc with Steve Horvath and just really kind of got immersed in the epigenetic clock world. But in addition to kind of my formal mentors, I've had great kind of colleagues and collaborators. So people like Luigi Ferrucci and Rafa Di Cabo at NIA, Jim Kirkland, who I've worked with a little bit at Mayo, and then obviously more recently, my work with David Sinclair and participating kind of on his reprogramming project, and then Juan Carlos Belmonte at Salk. So I think, I find the aging field to be very welcoming and friendly, and everyone's enthusiastic, and we all have the same goal. And I think, so far it's been kind of an amazing community to be a part of.
And a fascinating trajectory that you're on within it. So it's really, really great to hear about all this work, Morgan, and everything you're doing. It really an amazing set of programs. I wish you the best with all of this, because you're clearly on the cutting edge of what's happening. For everybody that is going to be listening to this episode on our podcast channels or watching on the YouTube channel, you've been listening to Dr. Morgan Levine, Assistant Professor of Pathology and Epidemiology at Yale School of Medicine, member of the Yale Combined Program in Computational Biology and Bioinformatics, and the Yale Center for Research on Aging. Morgan, I just want to say thank you for taking the time to know your schedule to come on our show. Thanks for everything you're doing there at Yale, and as we say on this program, thanks for helping to create the better tomorrow for all of us through this research. It's a very fascinating set of programs.
Thank you for having me. This was fun.